classifications are oxidoreductases, transferases, hydrolases, lyases, isomerases, and ligases. So oxidoreductases, like the name suggests, they kind of catalyze oxidoreductions, or oxidation and reduction reactions. And what happens is that these enzymes generally facilitate the transfer of electrons. They usually have a cofactor, which serves as an electron carrier, and you might have heard of these. These are NAD positive or NADP positives that are kind of commonly used in or seen in glycolysis and the pentose phosphate pathway. So those are kind of like the electron carriers that you'll see. The electron donors are called the reductants, whereas electron acceptors are called as the oxidants. So that's what oxidoreductases do. They transfer with electrons, and they usually just facilitate oxidation reduction reactions. The next one we're going to talk about are transferases, and what transferases do is that they take the functional groups from one group of molecules and transfer it to another group. So one example would be kinases. So kinases are usually responsible for taking a phosphate from ATP and transferring it to another molecule that needs it. So one example would be glucose to glucose 6-phosphate in the glycolysis pathway. This is catalyzed by a hexokinase, and what it does is it rips off a phosphate from an ATP and it adds it to the glucose at the 6th position, and it makes a glucose 6-phosphate. So now this glucose 6-phosphate molecule can no longer exit the cell, and it can go down glycolysis and give us energy that we need. Hydrolysis is the next type of enzyme, and what this does is it basically just breaks a molecule into two with the addition of water across the bond, and it kind of releases water in some cases as well. The next similar type of reaction are lyases. So these also involve cleavage of smaller molecules, but they do not involve water, so they use some other sorts of methods, other not involving water, to kind of cleave the bond. Isomerases, they don't really do a lot in terms of involving other kind of molecules. What they just do is they take a molecule, and if they see a bond, let's say at a double bond at a position between 2 and 3, they might just shift it to a position at 4 and 5. So they just like rearrange bonds and kind of deal with the same molecule throughout. Ligases, what they do are they're kind of similar to lyases in that they break down molecules, but they do it for much larger molecules. So they also don't require water. It's just a bigger molecule that they're going to be breaking down.